All right, so very warm welcome to all the participants. Welcome to this course, which is Basic Biology for Engineers, where we are going to focus mainly on the omics technologies. And this e-course is supported by TEQIP, third from the KIT project at IIT Bombay. So we are going to take this course for next 2 weeks. Especially it will be half a day every day this week and the next week, where we are going to give you some basics about how some of these biological concepts are crucial, but also we will try to make a balance that how engineering devices are required, how the big data being generated, and how that could be analyzed. So there are many aspects of investigating these kind of problems where you will see the basic concepts to the engineering applications both are equally important. So in this course we have various eminent speakers and instructors who are going to support this program, who are going to give you some basics and different type of hands-on activity. So today we will have Professor Siddharth Datta Gupta. He is a professor in Department of Electrical Engineering at IIT Bombay. Uh, we will have Professor Rodrigo Barderas. He's coming from Spain. He works essentially on the microarray technology. Tomorrow we will have Professor Graham Ball from Nottingham Trent University. He will talk more on the various ways of data repository analysis, looking at Array Express, how to utilize the data from the resources and make sense of them using machine learning and other kind of bioinformatic tools. On Sunday, we will have Professor Sanjeev Sabnis. He will talk to you about various basic mathematical terminologies like statistical tools, what is required for analyzing the data and making sure what we get is the significant information. And in the following week, we'll have again have some of these speakers, and one more new speaker will be added, which will be Dr. Geraint Morgan. He comes from Open University, UK. He will give you some more, his experience of making mass spectrometers, which is one of the very complex proteomics technology, and how that can be used for different applications. So as you can see, the program is designed in such a way that you will have a good balance of biologists, engineers, and people who are working on the cutting edge of these kind of protein-based technologies and omics and big data sciences they're utilizing for different applications. So, by the way, are you able to admit people who are coming, because a lot of people are keep coming joining the.
Yes, I am admitting all of them.
Yes, so please do that because very much interrupting my flow of presentations.
Yes, sir, you can continue. I will do admitting from here.
All right. Okay, everyone. So as you know, the session is going to be recorded, and also it is going to be attendance is going to be mandatory for all the sessions. So welcome to this particular course, Basic Biology for Engineers and Big Data Analysis. I'm going to start a very brief introduction lecture where I'm going to talk to you about why we need to talk about some of these biological concepts to engineers. I'm hoping most of you are the faculty members of different engineering institutes who are already working on the cutting edge technologies and you have a very specific research interest. But still our goal was to give you some insights about what is happening in the growing space of healthcare sector and how best the interdisciplinary minds could actually work together and try to solve the big problems. So the first part of this short presentation will talk about why we think biology is really important for engineers. And second, I will give you at least some idea for few DNA tools or technologies which are being used for the healthcare revolutions. So let us first think about why we thought about this course and why we think some of these biological concepts will be important for the engineers, right? Uh, I'm sure all of you encounter various issues on the day-to-day basis, whether it comes to pollution, global warming, looking at different type of health issues, and more recently, I'm sure everybody now appreciate the need for healthcare with the COVID-19 pandemic, various type of public health issues which are arising, and of course, on and off, you see various type of natural disasters different, the natural calamities which are happening, like cyclones come sometime, and many of these, issues, they leave long-term impact, which is, of course, does not require a single mind to work. It requires interdisciplinary skills to find the effective solutions. Like in COVID-19 case, you see, like, on one hand, you need the clinicians and biologists to understand about the pathogen. On other hand, you need the ventilators, you need devices, you need the tracking system. So you need engineers and you need the data tracking system. All of these skills together is somehow able to control and, to contribute something to combat this pandemic, right? So this is where I think the interdisciplinary nature of working together of biologists and engineers are really crucial. And I'm sure same example is very much valid for all of these various spheres which I have shown here. Many of you who are already quite advanced in your career, I'm sure you are aware, but still I would like to emphasize that there are huge projects going on from different industries, which are all technology giants, and they are actually investing heavily in the biology projects, right? For example, Google has different projects which are linked to biology, and especially Google Brain is one of the very popular project where they're looking at how they can look into the artificial intelligence, how one could understand that how brain works, and can you simulate those conditions, and you can actually make the Google Brain which can be working as intelligent as the human brain. Of course, there are other initiatives which is looking at how to even look at making the contact lens. A contact lens, I'm sure, is very old technology. It's not very new, but utilize the contact lens to understand what is happening inside an individual. So the physiological changes which are happening, how to try to reflect that change. And given that eyes have the fluid which is coming in the physiological condition of a given individual, if you have a contact lens which is in contact to that, you are actually able to all the time in the real time way, you can monitor the changes. And that's where some of the glucose sensors for the diabetic measurements have come forward. Now, another Google project is also looking at much beyond just the looking at glucose biosensors. And let me show you this clip from this link.
And in your bellies. Is this the future of your health? Google said this week that it's testing a smart contact lens that can help people manage diabetes, which affects almost 400 million people across the world, and that's expected to grow by quite a bit in the next couple decades. So it's no wonder people took notice when Google announced this lens, which can track your glucose through your tears. There's a tiny chip embedded on the lens that, if it works correctly, could eliminate the inconvenience and the pain involved in diabetes management. But Google's not the only one working on this type of technology. A team from Sweden designed a lens that has a fuel cell that can not only monitor glucose, but actually create small amounts of power. A Swiss company called Sensimed Triggerfish created a similar lens intended for glaucoma patients. And Google's version originated with University of Washington and Microsoft, which was once home to the lead researcher on the project. And other than the obvious medical potential, a lot of people have been speculating about what else smart lenses could be used for. The Microsoft research video from 2011 talks about the natural user interface vision of the lenses that do much more than just act as sensors.
Walking down the street, I could put people's names up, people that I recognize, people that I might want to recognize, information about them. The possibilities are endless.
Other clues to where tiny chips could take us? Well, the FDA has already approved a swallowable microchip the size of a grain of sand. Made by a company called Proteus Digital Health, it can be embedded in drugs and connected to a skin patch that sends info to your doctor on how you're responding. It can also sense physical activity, heart rate, and...
All right, so small devices with the understanding of biology, with the understanding of artificial intelligence, you can see that how much scope it has, and one could try to get a lot of information just by using these smart contact lenses, right?
And in your belly.
Likewise, I'm sure you have heard about IBM's initiative of Watson, which is taking a lot of knowledge from the literature and try to make this supercomputer, which is going to work like as a doctor, which is able to provide a lot of information. Of course, you will see the limitations of what human medical practitioners can think about as compared to the computer, but still it is really a very close simulation for that.
Important for Watson to generate a large number of possible answers and narrow them down from there, because if the correct answer isn't included during the initial sweep, There is no possible way for Watson to identify and justify the right answer at the end. Step 3. Hypothesis and evidence scoring. Of course, it's not enough for Watson to just come up with answers. It has to support and defend them. So after downgrading obviously wrong answers, Watson finds passages from...
All right, so I just gave you a glimpse that, Watson works very much in the scientific manner, the way we do experiments in the lab, and try to acquire a lot of data, build the hypothesis, and then try to make the prediction. But again, you can think about how all the giants, the big companies, they are trying to put investment toward understanding the various type of biological issues. And all the big institutes which you might have heard about, the MIT, Harvard, Stanford, University of California, all of them are heavily investing in biomedical and bioengineering aspect of research. Even IIT Bombay is quite leading various aspects of biology research. And the interface of engineers and the healthcare sector is, you can see immensely. We have almost 15% faculty members who are working very closely on various problems linked to the bioresearch, applied and the translational research activities. In Mumbai, we also have a healthcare consortium, which IIT Bombay is leading, where different hospitals, they're also partnering to this particular initiative of IIT Bombay. And they make this healthcare consortium, which is providing that ecosystem where you can start from the concept, do the experiments, and then validate your hypothesis and results in the clinical settings, and followed by take to the industrial partners for the follow-up translational activities of those. So again, there is that kind of ecosystem is required, and that mindset is required when you have to build that expertise of different individuals, different researchers, and try to work together in those aspects. So where we are leading towards? We are leading towards, of course, the COVID-19 pandemic has given a very clear message to the whole community. But overall, we are looking at a very new biology, which is not having any boundaries, which is going to look at different biological system understanding, which is going to be more based on the solutions which we want to find for various type of societal problem. Like right now we have COVID-19 pandemic. So you'd like to look at that as a problem and try to bring all solutions possible, from the sensors to the big data to various type of therapeutic aspect. All of them is required to really try to combat those problems. So we are talking about a paradigm shift in our thoughts, how the engineers and biologists, they used to work very much in isolation, but now we are looking at a new group of scientists who are working very closely on the problem and trying to bring with different interdisciplinary approaches. So in next segment, I will give you some flavor about how some of the recent technologies like next generation sequencing and DNA tools, they're actually being used to study various mutations in our gene, or how different genes are expressing, and those information could be important for the healthcare. So again, once you start looking at the genes, which is giving the blueprint of the life, and how different RNA or the proteins are being formed from those, these information gives you some clues about what is going on in the in an individual or any life science system. So this image shows one of the instrument platform known as next generation sequencer, which can sequence the genes or the nucleotides at a very fast pace. And of course, I'll talk a little bit more about this tomorrow, but this just gives you some idea that now we have, with so much engineering advancement happening in this particular area, we have very fast sequencer which can actually sequence all the genes of a human in just a couple of hours time. And that is the huge change which we see now as compared to 15 years ago, when even to just sequence one human, all the genes, it took almost 15 years time. But now after 2002-3, when that project was completed, which is Human Genome Project, now we have the rapid advancement in the sequencing technology of DNA. And now we have different versions of next generation sequencers, which is relying on different principles. And those are all physical principles trying to sequence the nucleotides rapidly. And now the cost has also come down very much. As a result, it can be directly now taken forward by the healthcare community. I'll just show you this news clip of one of the very modern example how understanding the mutation of a given gene could be helpful for predicting somebody's cancer, and also can we try to control that ahead of time. So this Associate Press clip, please watch that.
Her face is known around the world, and now her secret is known too. Angelina Jolie revealed in a New York Times editorial Tuesday that she had a preventive double mastectomy earlier this year, after discovering she has a gene defect that greatly increases her chances of getting breast and ovarian cancer. She writes, "My doctors estimated that I had an 87% risk of breast cancer and a 50% risk of ovarian cancer, although the risk is different in the case of each woman." While Jolie's genetic risk was very high, cancer experts say it's also fairly rare.
So 5% of breast cancers in the United States occur in women who have the genetic mutation, the BRCA1 or the BRCA2 gene.
Doctors say women with a family history of breast or ovarian cancer at an early age are more likely to carry the mutation, and insurance companies usually pay for genetic testing. For those who test positive, Dr. Rachel Brem says having both breasts removed, like Jolie did, is a very personal and difficult decision. It should be made with the help of genetics and cancer specialists.
It doesn't mean that that's the right decision for everyone. And the important thing to do is to have frank discussions.
Jolie said she went public because she wanted women in her shoes to know they have options and can still look and feel like a woman after a mastectomy.
Well, I think one of the reasons that we're seeing an increase in what we call bilateral or a mastectomy on both sides is that there are better reconstruction techniques.
The actress was treated in a California breast cancer center.
We hope that the awareness she is raising around the world will save countless lives.
Jolie's star power is already drawing attention to a complicated choice some women must face. Kelly Daschle, Associated Press.
All right, so you just watched this clip of Angelina Jolie, very popular face in Hollywood. And her family has a history of having breast cancer and ovarian cancer in her mom and aunt's cancer they had. And as a result, she was in the high cancer group that, she will have the high risk of the cancer. So one of the genes which is now known to be responsible for the breast cancer, like BRCA1 gene, they did sequencing of that gene from her and found that she has very high risk. The mutation was very high. And then she took a choice of mastectomy to remove the breast to try to prevent the possibility of, encountering this cancer at advanced stage. And that was, as you have heard in the clip, it was a tough decision, but look at the kind of awareness now which we have based on the sequencing and knowing about our gene and understanding that which can cause what type of problem and how best one could try to even prevent those problems ahead of time. Let me take you to another example, which is from a researcher. His name is Dr. Lucas Wartman. He's from Washington University in US. And he was working on the leukemia research. And unfortunately, he himself got that blood cancer or leukemia. So he wanted to sequence his own genome to find out which genes are actually having mutation. At that time in US, they were not supporting the gene sequencing as a part of their healthcare regulations, but university allowed him to do that. And you can watch this clip from the Barnes Jewish Hospital, what happened to Dr. Lucas Wartman.
Fortunately, during my last year of fellowship, again, started feeling a little bit more run down. It happened more suddenly this time. This past July, was diagnosed again with relapsed leukemia. Was admitted into the hospital and started on very strong chemotherapy. Unfortunately, though, the leukemia didn't go into remission after being treated with this very high-dose chemotherapy. There were no real promising treatments on the horizon, and so things looked rather bleak. But at the same time, as I was being treated, my leukemia genome was being sequenced by the Genome Institute here at Washington University School of Medicine. We got back the sequencing results, which showed that there were promising targets that weren't even in the scope of our standard treatment protocols for this type of leukemia. Because of those results, I was able to start less aggressive chemotherapy that I could tolerate, and then targeted therapy that had been approved for actually renal cell or kidney cancer. Within a few days, my blood counts improved, And within a few weeks, my leukemia was in remission. So it was really going from a situation that looked really bleak to now a situation where there was hope again.
All right. So in his case, when they looked at what is happening at his genome level, that what changes are happening in his because of cancer, they found one particular gene, which is FLT3 gene, FMS like tyrosine kinase 3, that was showing overexpression. And for that, there was no drug known for the leukemia treatment, but there was a drug known in the renal cell cancer, which is the kidney cancer. So they tried that drug, which is sunitinib, to try to treat the tumor in this case, and that actually worked well for him. So that actually emphasizes the need to know the right targets and how best that right target could be used for the therapeutic aspects. So, understanding these genes, their expression, and how they know the networks which is working together in our body for these genes at the entire genome level, and how some of the technologies can be used. One of the technology is the DNA microarrays. Is that what we are going to have the focus in our today afternoon session? And just because to connect you on some basic biology part here, our body is made of billions of cells, and every cell, one of these cells is shown here, contains all the genes which have information for all our various essential functions. So, for example, if you look at insect here, like from the brain or the nerve cell, it also contains all the genes. So as if you look at muscle cell, it also contains all the genes. Or if you look at intestinal cell, it also contains all the gene. As you can see, this is a very interesting reference from the NIH. So now all the cell contains all the gene, but all the genes are not expressing the protein in all the organs, because otherwise it is going to waste the cell's energy to make all the protein for every single gene everywhere. So in the nerve cell, you're only going to see the specific neurons are going to express, and only those proteins are going to be produced. In muscle cell, only specific proteins are going to express and produce. Intestinal cell, only those proteins will be produced, which is important for those functions. So now what you can see like here, that while the thread is same, but only specific components of those are expressing in different type of cell belonging to different organs. And those are termed as a gene expression, how the genes are expressing in specific organs, right, or specific organelle. So a technique known as the microarray that actually try to measure these kind of expression analysis. And some of the figures which I'm showing you is from a book known as Campbell, a biology book. So if let's say you have a normal individual and you are taking some sample from biological sample from that normal individual, or you are taking some sample from a cancer individual, and you want to measure that which genes are showing expression change. So now in this case, RNA was taken and labeled with fluorescence dye Cy3. RNA was taken, cDNA was made and fluorescence dye Cy5. Both of them are mixed, hybridized on a small chip. These are small glass slide, like microscopic glass slides, and then you have these oligos printed on those. And when now you are adding your patient samples on those, they are going to hybridize on the chip, and then because they have the fluorescent dyes for the normal Cy3 and for the cancer Cy5, then you are going to look at those expression, that what happens to every gene which is on my chip and their expression from the cDNA level. What is binding over there? Is that binding with this showing the Cy3 probe or Cy5 probe, or with the mixture of the two, and that means there is no change. So if a gene is expressed highly in the cancer cell, it will be shown with the red color. If it is highly expressed in the normal, it will be shown in the green color. I'm just giving you an example. It is not always the case. You can always do the dye swapping and use different dyes. You can always use the Cy3 for cancer cell or Cy5 for normal cell. But in this case, then now you are going to see 4 patterns. One is that if the gene is expressing high in the normal cell, that will be green. If a gene is expressing high in the cancer cell, in the red. If both of them are on the same spot, there is no change, then it will look like you are going to have the, there is no expression change, or you will have some empty spot which is going to just show you the probe and the there is no binding over there. So something like this can be so useful if you think about how our knowledge in the healthcare sector is evolving with these growing technologies. So breast cancer, as you just heard for Angelina Jolie's case, was considered to be one cancer type earlier. And all the women used to get the same treatment, same drugs, irrespective of what type of tumor they may have, of the subtype of tumor they may have, because we had no idea for that breast cancer is only one cancer or whether there are some subtypes of that. Eventually, when people looked at the gene expression profile and the mutations of those, they realized that the breast cancer is not one cancer, it is actually 4 different types of cancer. And there are 4 subtypes of that: luminal A, luminal B, HER2, and basal-like, given the type of gene expression patterns which is very distinct in these 4 different subgroups. So a given woman will belonging to either luminal A type or B type or HER2 or basal-like if she encounters the breast cancer. So here the estrogen receptor levels are very high, estrogen receptors are slightly low, ER level is negative in these 2 cases. Progesterone receptor is slightly high in both of these cases, negative in these cases. Now HER2 herceptin receptor gene, that is quite high in the HER2 group and absent in other groups. So based on looking at these gene expression signatures, now these subtypes have been made. And now what is interesting for you to know that each subgroup requires different type of therapy. So if you give the same drug to all the women, irrespective of which subtype of cancer they belong to, it's not going to work well with them. And they're going to see a lot of toxicity, the side effects, and that's why you see with the cancer chemotherapy that people show, the hairs are falling and immune system goes down and they see so much, pain during the treatment, because we are not able to give them the right, drug which can bind to the target, but rather every patient is bombarded with the drugs, overloaded without knowing what type of changes they have. And that's where some of these precision technologies are quite important for us to learn. So in the this week's segment, we are going to talk some of the DNA-based technologies, especially microarray-based technologies, which is both DNA and protein microarrays can be used over there. And you will see that you need good engineering interventions for making devices and a lot of data analysis skill to analyze the big data and make sense of the data. So broadly, first you want to make these microarrays which contains all the human genes or all the human proteins. For doing that, you have to do some biological experiment to do the protein purification or DNA, do the DNA purification, and then print those slides. Once you have printed those slides, then you can see those patterns in the image scanners. So again, a lot of emphasis is on to how to make good type of microarray-based image scanners, how to print these good slides, how to make good devices which can do the high density printing of the arrays. And then, of course, once you export the data, how to analyze the data. So just immediately after me, we are going to have 2 distinguished speakers, Professor Siddharth P. Datta Gupta from the Electrical Engineering Department of IT Bombay, who is, as you can see, he's professor from the Electrical Engineering Department, but he works on the interface of engineering and healthcare and biological aspects. And his group is also interested to look into the device fabrications for the microarray and other type of imaging applications. And then we will have in the afternoon Dr. Rodrigo Barderas from Spain, who will talk to you about the printing technologies and some of its applications. So that's all from my side for this